Okay. Good afternoon or morning, everyone, depending on where you're at. It's my pleasure to introduce Taylor Royalty, who is a C-DEBI doctoral fellow working on his PhD at the University of Tennessee, Knoxville with Andrew Steen. Taylor got his bachelor's degree in biology from Western Carolina University and a master's degree in atmospheric science from North Carolina State. That's really nice, Taylor, to see that you've developed a broad understanding of environmental science before diving into the most interesting area of all. So Taylor is doing really cool work using omics approaches to understand community microbial ecology with special emphasis on subsurface microbial communities. And with that, I'll hand control over to Taylor to begin your talk.
Okay, thank you. So before I get started, I really want to thank C-DEBI for funding my doctoral fellowship. It's been really great to have the opportunity to think about some of the ideas underlying this talk and some of the other research that I'm doing. So that's been really awesome. And so the title of my talk is Quantitatively Partitioning Microbial Genomic Traits Among Taxonomic Ranks. Implications for subsurface microbial communities. And so to start the talk, I kind of want to ask this question or kind of get at this idea of what I mean by the title of the talk, Quantitatively Partitioning Microbial Genomic Traits Among Taxonomic Ranks. And so I think the best way to do this is to start with an example. And so if you haven't seen this application, it's pretty cool. It's called an AnnoTree. It's available online, and so the idea behind it is you can look at the tree of life either from a bacterial perspective or an archaeal perspective. And the whole premise is you can choose conserved domains, and they got different types of annotation schemes, and you can look at the distribution of this trait that you've chosen across the entire tree of life. And you can choose different taxonomic resolutions, whether it be phylum, class, order, family, genus, species. And so the thing that's very apparent, or very quickly apparent, is that so in this case, the gray represents lineages that have an absence of this trait, whereas blue represents presence. And so you'll notice that there are certain lineages that are dominated, or these lineages are dominated by these clades are dominated by lineages which lack that trait, whereas others have the presence. And so this is a pretty cool observation because inherently there's a putative relationship between microbial phylogeny and traits, and this is kind of what it suggests. But in this case, though, lineage isn't necessarily a 100% accurate predictor of the absence or presence of trait. For instance, you can look at these clades that are dominated by these gray lineages, or they're missing the trait, but there are instances where certain members of those clades have that trait. And similarly speaking, you can look at, say, this one here, where most of the lineages do have this aminopeptidase N. There are instances that this trait is absent. And so it's often becomes convenient for us to make arguments where if we see certain lineages and environments, we say, oh, well, like we have some concept of the sort of metabolic processes that are happening. And so, you can think of, like, for instance, the cyanobacteria, the desulfovibronelles, and you can say, if I see a cyanobacteria in my environment, well, I know that there's oxic photosynthesis going on, and so it's likely an oxic environment. Or maybe if you see these guys, you see some sulfate reduction, and so you're in an anoxic environment. And so it's convenient to make these arguments. But sometimes it's also convenient to make arguments and to contrast this, which is, for instance, if you look at the Rosiobacter, where you have a number of very closely related lineages, but their metabolisms are very different. Some are oxidizing nitrogen, some of them are performing photosynthesis, and so it's convenient for us to say, no, lineage doesn't tell us anything about function. And so the reality here is that it's some intermediate, right? Like sometimes lineage is very good as a predictor and other times it's not. And so we want to quantify this. And so that's kind of the underlying idea of the work that I'm about to present, is that we are quantifying the relationship between taxonomic groups and the underlying traits within their genomes. And so the reason that we ultimately care about this is because as people, we really care about the sort of processes that are driving dynamics in the environment. These are processes that, for the most part, we care about, whether it's biogeochemical reservoirs, whether or not it's human health, looking for industrial applications, medicine. And so oftentimes these processes are measurable, but sometimes they're not. And these cryptic processes are often the result of the sorts of metabolisms that we can't measure them because the concentrations of the constituents are too low or the reactions are too quick. And so if we had some ability to gain insight on these processes, that would be useful for us to understand the interaction between microbial metabolisms and environment. And so this requires a more holistic picture of microbial ecology. And so this is kind of my conceptual diagram of microbial ecology, but there's this interaction between lineage and environment, and there's this interaction between lineage and metabolism. And so if we had some sort of constraint on the relationship between what microbial metabolisms can do and the lineages, and then we also had some notion of where lineages are in respect to environments, then ideally, we could maybe make some inferences on what these lineages are doing in these environments. And so one of the prerequisites to this is quantifying the relationship between lineage and metabolism. And so, as I said before, it's not a 100% predictor, right, lineage, as far as predicting metabolism. And this is in part because there's a lot of evolutionary processes influencing the traits that are in genomes. You have things like horizontal gene transfer, either viruses introducing genomic material into genomes, or you have recombination, you can have genetic drift, mutations. So there's a lot of processes operating on genomic traits. So this relationship needs to really be constrained quantitatively. And so before I really get into the results of the work that we've done, we really need to think about how we think about lineage, define lineage, and how we think about metabolism, define metabolism. Because like most things, there's a lot of different ways we can do it, and there's ramifications and implications from how we choose to define these things. And so to start off with, I'll kind of talk about this lineage. And so over the past 20 years, there's been a variety of different taxonomic systems proposed. NCBI and Silva are some of the more common ones that have been used lately. And so what I'm really going to say about this is that these lineages or these taxonomic systems are not sufficient when trying to constrain the relationship between lineage and metabolism. And this is due to some underlying issues. Particularly lineage, there are instances when defining taxonomic groups, they're tied to phenotype and morphology. And so you can think of some examples like the Clostridium, for instance, where the whole fact that this group even exists as a name is because these organisms kind of do similar things, but there's not really a evolutionary justification for calling them that. And so this is a problem because if we're trying to quantify the relationship between lineage and metabolism, and lineage is partly defined, or that the taxonomic group is partly defined by metabolism, you kind of get into this circular argument. And so this last year, the Genome Taxonomy database was proposed as or GTDB was proposed as a new taxonomy. And this was proposed by Parks et al. And the Hugenholtz group. And so this was really a revolutionary idea, because what's going on now is we are algorithmically defining taxonomic ranks, or we're assigning, so the taxonomic ranks here are being algorithmically defined. And so relatedness here is defined purely based off of sequence alignment of single copy marker genes. And so in the case of bacteria, there's 120 marker genes that are used. In the case of archaea, there's 122. And so relatedness, and so these traits are well conserved with lineage, and they're not necessarily related directly to function and metabolism. A lot of them are like things like ribosomal proteins. And so what they do is they say, okay, look, we're going to standardize the distance that they're going to standardize the definition of ranks. And so they do this using this relative evolutionary divergence metric. And so they say the last common ancestor here, our last universal common ancestor gets a value of 0, and then extant taxa get a value of 1. And then what they do is they define taxonomic ranks at these set intervals. And so this is derived based off of bootstrap analyses, these median values. But the idea here is that this is an algorithm that's assigning group names based off of a metric, the concatenated protein sequence alignments in terms of defining relatedness. And so you'll notice here though, so up here is NCBI and down here is GDTP. You'll notice the different colors, and so the red corresponds to polyphyletic groups as defined by these concatenated protein sequence alignments. Yellow is this operational monophyletic, and green is monophyletic. And so the big point here is that if you look at how GDTP defines taxonomic ranks based off these standard intervals using their protein sequence alignment defining relatedness, this is a very different sort of taxonomy than the NCBI. And so what's powerful about this, again, is we can now test ideas related to how microbes are, their evolutionary history to function. And so this is critical. So we adopt GDTP taxonomy for the work that we're presenting here. The next question here is how we choose to define metabolism. And so we kind of need to think a little bit about how we come to describe metabolism of organisms. And so ideally, you could even argue this is the best way, is pure culture, where you find a microbe, you isolate it, you grow it up in culture, and then you can do things like modulate, nutrient concentrations, you can knock out genes, and then you observe some phenotype, and then you go down the list of all the genes that you can do these for without killing the microbe, and then you've characterized the metabolism. An alternative approach is one that's algorithm-based approach, where you can sequence a genome and then identify genes within that genome, and then you compare it to a reference database of genes that have known function, likely related to pure culture experiments. And then the question is, which of those genes matches the gene that I have? And so again, this uses sequence alignments. And so when you get a match, you give the annotation from the known gene to the unknown gene, and you say, okay, this has this potential to perform this function. You don't have any information about expression, but you know a little bit more about the organism at hand. And so unfortunately, for the most part, we're left to utilize the second approach, because a disproportionate number of lineages do not have cultured representatives, and it makes it challenging if we were to just rely on this as a technique, then there would be a lot of lineages we couldn't say anything about. And so it doesn't stop there, though, because ultimately there's a variety of different ways we can annotate genes. And so what I'm showing here on the left is a relationship between how, and so the functional form of this is not necessarily true, but it's more so to demonstrate the general point. But what we're describing here is the relationship between our ability to describe something generally, or how comparable it is across different lineages, and the amount of specificity or how much information we can say about the function of that trait using that annotation. And so the idea here is the more comparable something is, the less we can say about it. And then conversely, the more we can say something about something, the less comparable it is across multiple lineages. And so you can think of something down here like a protein sequence alignment between two genes from two strains, and they're the same gene. And so you can learn something about maybe about why the reactivity of, or the rate of reaction from some hydrolase is much quicker in strain A opposed to strain B. But if we think about this in the context of distantly related lineages, such as cyanobacteria and sulfovibrinales, the metabolisms of these organisms are very different, and the genes that they have are completely different. And so, although you can do a protein sequence alignment of completely unrelated proteins, it's not going to of completely unrelated proteins, it's not really going to mean anything. Now, that doesn't necessarily imply, though, that the phenotypes of these two organisms is completely uncomparable, right? Because ultimately, cyanobacteria are using photosynthesis as a means to obtain energy, and similarly speaking, sulfate-reducing bacteria use sulfate as a source of to generate energy. So these phenotypes are comparable at some extent, and so we rely on more general means to describe the functionality of their traits, such as assigning genes to carbohydrate metabolism, lipid metabolism, amino acid metabolism, etc. And so a consequence of this is we no longer have any sort of notion about substrate, right? We no longer know that CO2 is our terminal electron acceptor. We no longer know that sulfate is our terminal electron acceptor. But we can compare these two seemingly very different lineages. And so for the work that we use a somewhat intermediate annotation scheme. It's a cluster of orthologous groups. There's about 5,700 protein annotations. And so the problem with this number here is there's actually more protein annotations than there are typically genes in a genome of a microbe. And so we end up, when trying to do comparisons, we end up with a very sparse data matrix doing comparisons. There's a lot of absences still. And so this makes statistical analyses a little tricky. And so what we end up having to do is, which is convenient, is there's 25 broad metabolic categories which these protein annotations can be mapped to. And we can tabulate the abundance of these 25 broad metabolic categories. And this is quantifiable. And so just a little bit more on the topic of COGs. We utilize reverse position specific BLAST algorithm. And all this does is it takes we have a model sequence for a protein annotation, and then we compare it to a gene, and there's some confidence interval, and if the confidence interval is good enough, then we can assign the annotation associated with that model to the name of the gene, or associate that annotation with the gene. And then we know how that maps to these broad metabolic categories. And so what we're measuring is, so to kind of give you an idea of what we're measuring, if you look at the circle that kind of represents some microbe's genome, some fraction of the genes within its genome are being assigned to these different categories, and some component of these are actually going to be unassigned as well. And so, like I said, there's 25 COG functional categories. We utilize 23. 2 of these aren't particularly relevant to prokaryotic organisms, so they weren't included. And what we see by these different categories, though, is, again, there is some information about functionality, right? Like we can conceptualize what cytoskeleton means. We can understand chromatin structure, but we lose specificity. So you have cell cycle control. So you can imagine a lineage with elevated amounts of cell cycle control. And you could conceive a scenario, for instance, where maybe the high abundance of cell control COGs is to help upregulate cell division. And conversely speaking, it may actually suppress cell division. And so you end up with the opposite phenotypes. So we lose specificity at the expense of comparability here. And the last thing I kind of want to talk about this is we're not actually, so for our analyses and our comparisons, we're not actually comparing the abundances of these different categories within a genome. What we're comparing is the relative abundance of these categories in respect to what we would expect for a genome of equal size. And so I'm not really going to talk much more about that, but we talk about it in the paper on this. So if you can check that out if you're interested. And so kind of going back to the central question here, we're really interested in quantifying this relationship. We're defining lineage using GTDB, and we're defining metabolism using COG functional categories. And so, like I said, this was actually put up online just last week. We published this in Hemisphere, so if you're interested, check it out. We tried to get our hands on as many genomes as we could. And so genomes kind of used loosely here for simplicity, but we got about 10,000 metagenome assembled genomes from GenBank, high quality. These represent uncultured lineages. We obtained metagenome assembled genomes from JGI, IMG. We also got single amplified genomes. And in addition to that, we downloaded the entire RefSeq database. I think it was RefSeq 92. And so what we did was we assigned GTDB taxonomy to each of these, and then we calculated the abundance of each COG functional category within each of these genomes. And so the problem with this 130,000 number is most of those genomes are associated with model organisms. So a lot of them are E. coli, a lot of Salmonella, a lot of Pseudomonas. And so if we're doing statistics on this, we end up with a very unbalanced experimental design. And so what we ended up doing was we averaged the COG functional category profile for these individual genomes for all genomes that were of the same species. So there was only one profile representing E. coli, one profile for Salmonella, one, etc. And then what we did was we excluded any genera that did not have at least 10 unique species. And the reason we needed at least 10, which was a little bit of an arbitrary number, but we need to do statistics and we need replicates representing genera. And so in total, we were left with 13,735 unique lineages for our analysis. We performed a principal component analysis on that using those COG functional category profiles. Again, these are relative abundances, not abundances, because we normalize. And what I'm showing here is I have these different panels, and the black lines contours are the same in each of the panels, and those just represent the density of all the lineages, and I'm just showing the top 10 most abundant lineages. And the whole point here is these different colored contours correspond with the lineage names above, and their center of mass is just occupying different space in principal component space. And so this is some indication that there is some pattern and a relationship between lineage and COG functional categories. And so to kind of back up, you can really ask this question, well, how well do COGs actually represent all the genes within genomes? And so what we did was we looked at the fraction of genes that were assigned a COG functional category. And so what we saw was about, on average, 85% of genes were assigned a COG functional category. Some lineages were more well represented, such as the proteobacteria, whereas some were less well represented, like Archaeota. But overall, we thought this was a pretty good coverage, and things were pretty comparable. And so now I'm going to talk about the actual quantifying results, or the quantitative results. I'm not really going to go into like the specifics of the statistical analyses, but I will talk about the results from those. And so the first thing that we did was permutational multivariate ANOVA. On the y-axis, we have variance explained. On the x-axis, we have different taxonomic ranks. And so the whole question here is, how well does taxonomic rank explain the structure of functional categories within genomes? And so you can see here that the relative ratio in this little cartoon example of these different categories is different. And how well does taxonomy explain those differences? And so genus explains 41% of the variance. And so genus in itself is the best predictor. But the nice thing about this technique is we can actually attribute what fraction of that 41% variance is attributed from higher taxonomic ranks. Because if you're within a genus, inevitably you're in a given class, order, class, phylum. And so we can partition out that variance explained. And it actually turned out phylum had the highest explanatory power, followed by order, then genus, family, class. And so this was a bit surprising and interesting. And I will say all of these were statistically significant. And so given the fact that phylum had the best explanatory power, we were curious about how much diversity exists on phylum level across all phylum. And so what we did was we calculated the diversity of each phylum. And so how this was done was we defined a mean, there's a mean COG profile. So we have 23 variables, each representing a COG, and we defined a mean value. And then we measured the distance that individual species were within that phylum to that centroid. And so what we found was the Archaeota were the most diverse phylum. And then we saw this kind of sharp cutoff, or not a cutoff, but at this plateau where a bunch of lineages had pretty comparable levels of diversity. And then we saw this then we saw this drop-off in diversity. And so this drop-off in diversity was actually something of interest to us because we were curious if this was biologically relevant or a biological signal. And you could envision a scenario, say with the synergistota, where perhaps you have a phylum full of synergists, and so they have small genomes, small core genomes that are highly streamlined, and so they don't have a bunch of traits that can evolve very easily without messing everything up, and so you would expect a phylum with low diversity. Or does this reflect some sort of sampling bias? And so what we did was we plotted diversity of the individual phylum as a function of number of species within that phylum. And so we saw this asymptotic feature where low species had, or phyla with a low number of representatives had lower diversity, and then as you increase the number of representatives, you found more diversity up till about 100 lineages, and then you start to plateau. And so the implication here is, in some theoretical scenario, if we were to find more lineages or species of these guys, we would expect higher diversity. But this doesn't necessarily imply that there are, in fact, more representatives of these groups. So we wanted to go on and we wanted to contextualize the observations of these relative abundances for these different COG functional categories in the context of the evolutionary history of these microbes. And so the heat map here corresponds to relative abundance, and then we have the different COG functional categories along our x-axis over here, and then we have the tree of life along our y-axis. And so you'll probably notice that there are tons of bands of solid colors, for instance, right here in the proteobacteria. And so you see these clades of solid color, and what this indicates is that the relative abundance of that given COG category is conserved within that clade. And so you see bands on all sorts of resolutions. You know, there's some on the domain level, you got some on phylum level, class, genus, they're all over the place. So it's pretty cool. And it indicates that, you know, certain COG functional categories are being conserved in different ways. And so what I really want to get, and we talk about this a little bit in the paper too, we try to rationalize some of the observations and say like this clade right here where there's a lot of depletion across the board. But ultimately what I want to get at here is that what this represents is a model of metabolism for different genera. Each one of these terminal nodes represents a single genus. And so we have a profile of the metabolism for this genus. And so kind of going back to this central question here of to what extent does this relate, we quantified this. And I didn't present all the statistics that we done. We did some additional analyses in the paper, so check it out if you're interested. But what we're left with is a model of metabolism. So going back to what we want to do, which is make inferences, this is useful. And so we're kind of left now, if we want to do that, we got to constrain this relationship. And so this is kind of the second part of the talk where I'm going to talk about subsurface microbial communities and some inferences that we make. And so fortunately, somebody else has taken the time and energy to do this, the Earth Microbiome Project. Some of you may be familiar with, but this was published just a couple of years ago, Thompson et al. And what they do is they take 16S amplicon sequencing profiles of, I think it's some like 27,000 different microbial communities, various different environments from soil, sediment, freshwater lakes, saline lakes, you know, et cetera, et cetera, et cetera. And so what they show is the biogeography of these different genera is not random. You see structure in this data. And so this is great because we can couple our metabolic model between metabolism and lineage with their model of lineage environment. And the big thing here is that just needs consideration is this is based off of 16S data. And so you can't assign GTDB just based off 16S, but we have 16S data in our genomes, so we can fish out those, assign a Silva taxonomy to both of these, and then map our GTDB taxonomy to that Silva. And so what we wanted to do was, and I don't mean to throw this equation at you, but what we want to do is we want to measure a community level shifts and metabolism. And so what we can do is we can scale the relative abundance of a given lineage against the relative abundance of individual COG categories. And then we can sum those up to get a COG profile for that community. And so that's what we did. And so again, the idea is if we know what lineages do and we know where they're at, we can make inferences about what they're doing. And so the central question we tried to get at here is, are there differences, community level differences in metabolism for subsurface communities? Like, are all subsurface communities the same? Are they different? How different? And so the Thompson data set had over 1,000 samples from 12 different studies for subsurface sediment communities. And the thing that I want to say here is that we evaluated this on the class level. So we only had about 450 model metabolisms. Silva database has, I don't even know how many genera they have. They have far more than 450. And so if we were to just rely on that 450 genera model, we would end up not having a good characterization of those communities. And so what we had to do is we had to downsample our genera level model to higher taxonomic ranks. And so when we did it to a class level, we had a model metabolism for about 95% of the community. And so we performed, each of these data corresponds to, data points corresponds to a single community. And we performed principal component analysis on the COG profile for that individual community. And we found that the first principal component explained 44% of the variance, second explained 24% of the variance. And so if you've done these types of analyses before, that's a pretty substantial amount of variance explained by the first two principal components. And so the nice thing about the Thompson data set is they do publish chemical data. Unfortunately for this, and this chemical data can include things like sulfate, phosphate, nitrate, nitrite, oxygen, pH. But the problem here is that the sediment communities, a lot of this data was missing. So if we want to try to better understand this pattern here, we need to get creative. And so one idea we had was to look at the abundances of gamma proteobacteria and beta proteobacteria. And so this isn't necessarily specific to sediment communities, but there seems to be some evidence that these two lineages tend to be more abundant in copiotrophic environments, environments with high nutrients, high energy. And so if you believe that, you would argue that these communities over here are higher nutrient, higher energy. These down over here are lower nutrient, lower energy. And so the gray corresponds to complete absence. And so clearly there is a spatial pattern in this. The next question we had was, what's the redox state of these different sediment communities? And so the way we could get at this was looking at the absence and presence of certain marker lineages. So we looked for the presence of methanogens. And if there was the presence of methanogen, then you were colored blue. If there was no methanogen and there was a sulfate reducer, we colored you red. And if there was neither, then we colored you green, or not green, red. And so red maybe corresponds to oxic. But ultimately, you see this large discrepancy between the communities containing methanogens and those for sulfate-reducing bacteria and the oxic. And so this is very impressive split. And we were curious about what sort of COG profiles were driving this differentiation. And what we actually found was these communities that had more negative values on the first axis actually had enrichments in almost all COG functional categories. And so if you think about it, the way that this could happen is if across the board you had more genes that were not assigned to COG functional categories. And so the idea here is if 100% of your genes are assigned to COG categories, if you average the relative abundances for each of those COG categories, you should end up with a 1. But if not everything is being assigned to a COG category, then you end up being deplete on average. And so this would suggest that methane communities that contain methanogens have less genes being assigned to COG functional categories. And so maybe this suggests some novelty, or just the definitions of these categories don't work particularly well on methanogen communities for whatever reason. And so the last thing that we looked at was diversity. And we found that these methanogen communities actually appear almost maybe even more diverse than these oxic and sulfate-rich communities based off Shannon index. And so if you think about it, this would actually also imply that the observations that we saw about this depletion and traits, this is not just a single individual dominating these methanogen communities who happen to not have their traits being assigned to COG functional categories, but this is actually a community level shift in you know, these members being deplete and the assignment of COG functional categories. So that was an interesting observation. And so with that, I just want to say some concluding remarks. We quantified the relationship between lineage and microbial metabolisms using COG functional categories and GTDB. We found phylum was the most powerful predictor of metabolism, you know, and this was kind of contrary to some of the conventional wisdom in the papers that I had read, and I cite these in our paper, but you know, it seemed like prior analyses that were kind of focused on constraining this relationship, they tend to see a cutoff, that loss of signal at taxonomic ranks higher than order. So the fact that we saw phylum being the most powerful predictor with GTDB was cool. We coupled our metabolic model to 16S amplicon sequencing, which we feel may be a good way to get more information out of 16S sequencing experiments. And then lastly, we saw some interesting observations in the methanogen communities. Maybe there's some novelty in some of the traits, who knows? And so acknowledgements, I do want to thank Chad Burdyshaw from GIX. He helped us get some of the genomes from JGI-IMG. Anonymous reviewers, these guys were all stars. They were the most clear and constructive comments I think I've ever had. And then NSF for funding Drew as a part of this work. And I want to thank C-DEBI again for funding my doctoral fellowship. So with that, I'll take questions.
Oh, thank you, Taylor. That was great. We very much appreciate you sharing your research with the community. Yeah, so we have some time to take questions. If the audience could use the chat module. I don't know if it's showing up for everyone, but there's a little chat bubble at the bottom of the screen. And yeah, we'll take a moment to see if we have any questions.
Yeah, as far as the, okay, so Joy is asking if there was any enrichment in unclassified COGs in uncultured lineages. So I don't know if there was that's a good question. I don't know if there is less COGs being assigned for uncultured lineages. Well, actually, no, I do know the answer to that. The answer is no. So in general, uncultured lineages appear to be very similar to cultured lineages. We have a supplemental figure in our paper that shows that. Actually, no, we didn't publish that figure, but we did do the comparison. And when we did the Permanova analysis, it was significant, but the variance explained was something like 0.001. So no, I don't think so. You made some, but I'm sorry. No, I didn't check on the IODP websites. That's a good idea. I tried going through some of the, so some of that data was actually published. Like I know one of those papers was from Jennifer Biddle on some sequencing stuff that she had done, and I was able to find stuff like that. But that's a good idea. This is something like, there's only 12 studies with the samples that I was showing, so I'm definitely wanting to kind of comb through more papers. Given that the databases online, they're not very good with annotations, so I was trying to find more studies to include, but the way that they annotate them with the 16S data, it's like, I don't know which genes you're amplifying. So I'm probably gonna go through all the C-DEBI papers and try to find 16S sequencing experiments and then use those. But yeah, IODP, okay, I'll look through those. The combo of catabolism, and can you say something about predicting catabolism? I don't think so. So the question is, can we differentiate between catabolism and anabolism? I don't think so. So we can look at, like I had said, there's 5,700 COG domains, or yeah, protein domains, and we could maybe make some inference from those because those each have their own annotation, but from the metabolic categories themselves, it's a bit ambiguous, which is one of the big issues with this approach. But I mean, it's the other thing I view as strong evidence for this approach too, is the fact that we see signal still it's pretty impressive, but I don't think you can. Okay, do the COGs only represent functional similarities, or do you see similarities in protein sequence structure within groups? So we have not compared the actual protein sequences for the underlying proteins. Computationally, I don't know how difficult that would be or what we would look at. What I will say though is that Drew has, in a project I've kind of been fringely been involved with, he's been doing some machine learning where he's been trying to feed, and we're working with some people in the computer science department in our at our university, but where we're trying to predict annotation of just pure protein sequences and nucleotide sequences, and they've had some reasonable success with that. So that'd be an interesting comparison to see if there's some pattern and run the analysis on the underlying proteins in our genomes with the COG functional categories. Let's see. Okay. Cool. Is there any more questions? Preprint coming in. Cool. Okay. Preprint is coming. Didn't know that. Thanks, Joy. Well, like I said, guys, the paper is up. I did skip on some analyses. I actually have one of them here just to show you, but we also did quantify. So we did the permutational multivariate ANOVA, which again is kind of like a structure, but we could also quantify how much variance is explained of individual categories by taxonomic ranks using a variance component model. So this is just a regression or a intercept model, and you can. And so we do report these results as well, and it's kind of interesting just seeing how much variance is explained. Like for instance, coenzyme trafficking is explained by the phylum, right? Like a significant component, whereas cytoskeleton is nothing. So we published that data as well. So that's kind of another analysis we did, but
All right, well, thank you, Taylor, and thanks everyone who has tuned in to the series. For those who missed this talk, this will be uploaded to our YouTube channel shortly. And then coming up in about a month, October 3rd, our next speaker will be Dr. Jessica Labonte from Texas A&M Galveston. But thank you, everyone.
Yeah, thanks guys for tuning in.